Hello, today's topic is biochemistry questions. It is from biology. Let's discuss some questions. Here, first question is the Km value of an enzyme is best defined as the substrate concentration at which. We have four options. I have already mentioned the right answer. Right answer is option C, that is reaction velocity is half of Vmax. So let's discuss the explanation. The Km, which is Michaelis constant value, is defined as the substrate concentration at which initial reaction velocity is exactly half of the maximum velocity that the enzyme can achieve. And we can represent this, that is V is equal to half Vmax. You can say C Km, that is substrate concentration, that is half of Vmax. Okay, let's move to next question. Next question is which amino acid is most likely to be found at active site of enzymes involved in acid-base reaction. It also maintains the pH in the body, that is histidine. You just remember this answer. Explanation. Histidine is the most likely amino acid to be found at the active site of enzymes involved in acid-base catalysis. Question number 3, competitive inhibitors. Competitive inhibitors are those inhibitors which compete with the substrate so that it can decrease the enzyme reaction. Four options we have given. I have already highlighted the right answer, that is option B, increase Km without affecting Vmax. Competitive inhibitors bind to enzyme's active site, competing directly with the substrate. Because the inhibitors effect can be overcome by a sufficient high concentration of substrate, the Vmax, maximum reaction velocity of the enzyme reaction, remains unchanged. Number 4, the major regulatory enzyme of glycolysis is phosphofructokinase 1. Okay, explanation. Phosphofructokinase PFK1 is considered the major regulatory and rate limiting enzyme of glycolysis because it catalyzes the fast irreversible committed step of pathway, ensuring that the molecule produced fructose 1,6-bisphosphate proceeds through glycolysis to generate energy. Its activity is highly regulated by cellular energy levels and hormonal signals. Number 5, which of the following is an anaplerotic reaction? Anaplerotic reaction is the filling up reaction. Answer is pyruvate is equal to OAA, that is oxaloacetate. Anaplerotic reaction are filling up reaction that replenish the pool of intermediates in the citric acid cycle or TCA cycle when they are run off for biosynthesis of other molecules like amino acids or glucose. The conversion of pyruvate to oxaloacetate via the enzyme pyruvate carboxylase is primary anaplerotic reaction in many organisms, especially mammals. This reaction replenishes oxaloacetate, which is essential for TCA cycle to continue by condensing with acetyl coenzyme A to form citrate. Number 6 is the reducing power generated in pentose phosphate pathway is mainly fatty acid synthesis. You just remember this kind of question. Okay. Next question is which lipid is major component in a biological membrane, that is phospholipids. Okay. Which bond stabilizes the alpha helix structure in proteins? Hydrogen bond. Which enzyme is responsible for removing RNA primer during DNA replication in prokaryotes? That is DNA polymerase 1. DNA melting temperature, that is Tm, increases with an increase in GC content because GC content has triple bond, hydrogen bond. That's all about the question. In my next video, I will bring more questions. Thank you.